Today we'll talk about a paper that deals with LRP1. We've seen this receptor before, and tau endocytosis. So far we've seen LRP1 in the context of moving A beta peptides through membranes. It's generally a good thing because LRP1 is the one that moves A beta from the cerebral spinal fluid, so the liquid that bathes neurons and glia, into the bloodstream so that it can be taken away and metabolized, say, in the liver. Right? So LRP1 so far had been a really good thing. But today we'll see a paper that sort of opens up the possibility that LRP1 expression can contribute negatively to the progression of Alzheimer's disease via facilitating tau endocytosis. So the paper that is also uploaded in Canvas is tau uptake by human neurons depends on receptor LRP1 and kinase LRRK2. It's published in EMBO Journal last year. This is a pretty recent one. And what the authors mentioned in the introduction is the current model for how tau contributes to Alzheimer's disease is that tau is released from diseased neurons or dead neurons that burst out and release all of these tau proteins they had in them. And that released tau can be uptaken by nearby or neighbor synaptic neurons from those diseased or dead neurons by microglia. And that this accumulation of tau inside of neurons can serve as nucleation centers for further aggregation. Right? So the idea here is that all of these extra tau that neurons that are right next to dying neurons take up can serve as nucleation centers for these tau to accumulate and form those neurofibrillary tangles that make neurons so sick and eventually die. One thing that will be important in the context of this paper is that both monomeric and oligomeric tau and also fibrillar forms of fibrillar tangles of tau protein can be picked up by neurons. So it's not only the monomeric tau molecules that get endocytosed by neurons, all three forms can be endocytosed. But as we will see in the paper, not through the same mechanisms. So, and that's probably one of the strongest messages from this paper. What do we know about uptake of tau so far? Well, what we know is that heparan sulfate proteoglycans are needed for uptake of fibrillar aggregates. And one thing that I didn't write down here, I forgot, but the other thing that we know is that LRP1 has been observed as a receptor that can facilitate endocytosis of monomeric tau. So it seems like when tau is forming these neurofibrillary tangles that may have formed in these sick neurons before, and then when they died, got released into the extracellular medium, those tangles get picked up through heparan sulfate proteoglycans, whereas monomeric tau gets picked up by LRP1. So this is a new idea, new concept that neurons that are next to dying neurons can pick up tau and make things worse for themselves. So what the authors wanted to do is run a genetic screen to try and figure out what genes and pathways may be involved in these processes. And for that, they used a CRISPR approach that is a little different from the one that we discussed before. And as their sample or as their material, research material, they used iPSCs or iPSC-derived excitatory neurons. I'm sure that many of you may have heard of iPSs before. If anyone has, you guys want to share what you think they are, how you would describe them? Induced pluripotent stem cells. So that's a mouthful. So what do you mean by that? That's exactly right. So you're saying reverting back. So the idea here is that most cells in a grown organism are not pluripotent. Right? Most cells in a grown organism, for the most part, they can't even divide, right? And if they divide, they will most likely make a new copy of themselves. as that's sort of the idea that's been ingrained in us forever, that when a cell divides, two cells are formed that are identical to one another, two sister cells, right? So the classical idea of mitosis. But what's implied in that is that when a cell divides, it will give you a cell that is identical to itself. It doesn't have any pluripotency. It cannot become something else, right? So pluripotent stem cells, the naturally occurring ones, where do we find those? Where can you find a natural source of pluripotent stem cells? In the somatic, germinal is different.
Sorry, did you read the question?
Yeah, the question is, where do we find normally pluripotent stem cells? In the bone marrow.
I would say bone marrow
And thyroid, bone marrow. So those stem cells that you find in the bone marrow, are they pluripotent? Can they give rise to anything? Can they give rise to muscle or a neuron or an intestinal cell? What kind of cells will bone marrow stem cells give normally? B cells, right? So those are immune cells. What other cells? Eventually, the cousin of B cells, T cells, red blood cells, macrophages, monocytes, all blood cells. So bone marrow stem cells will give blood cells. Intestinal stem cells will give intestinal cells. Muscle stem cells will give muscle cells. And we can go on all day. But all of these are examples of, in some cases, even monopotent stem cells. They will give rise to only a type of stem cells. So where can we find truly pluripotent or totipotent stem cells? Where would you find a cell that can give rise to any organ in the body? In the umbilical cord, embryo, two sides of the same coin, right? So yes, you can sort of get some pluripotent stem cells from the umbilical cord, but more importantly, you can totally get them from the embryo. And we call those embryonic stem cells, and those are the ones that really, as long as you give them the proper directions, will give you anything. Will give you bone cells, neurons, intestinal cells, muscle, whatever. As long as you know what instructions to give those cells, you can generate anything out of them. So back to the iPSs. You mentioned that you revert back to pluripotent stem cells. That's exactly the point. In iPS technology, what we're taking is a cell that is committed to a cell fate and forcing it to reverse. We're inducing it to go back to that pluripotent stem cell. We're sort of turning back the clock on, or the developmental clock on that cell, such that that cell may have been a skin cell, may have been a blood cell, may have been a myocyte, a muscle cell. And what we're doing is we're saying, no, go back to the time when you could become anything. And those are iPSs, induced pluripotent stem cells. Did that make sense? Yeah, sorry, you missed it, but I couldn't upload the slides to Canvas, so we'll go old school. All right. So these guys will take these induced pluripotent stem cells that can become anything, instruct them to become neurons, and then conduct their genetic screen for genes that are important for this uptake of tau. So the approach is depicted here in a cartoon manner. If this image kind of rings a bell, that is because we've seen a very similar cartoon schematic of the screen approach in the CRISPR-I paper, right? So here they start off of a culture of human Cas9 expressing cells. And So they're shown here as a culture of cells that are not nothing yet, right? And why it is important that they express Cas9? Because what they're doing is these cells already have Cas9, and what the authors did was incubate those cells with a collection of viruses, each of which had a specific guide RNA for bringing that Cas9 to somewhere in the genome. So the collection of guide RNAs that they used would target genes in the human genome, more specifically in protein coding regions, right? So the cells already have Cas9, and what we're offering them is the guide RNAs that would take that Cas9 to a specific location in the genome. Where in the genome? Well, that depends on the guide RNA that Cas9 partners with. So the question becomes what guide RNA each cell gets. And if you guys remember in the CRISPR-I paper, we had seen that they flooded the cells with viruses because they wanted actually many guide RNAs to go into each of the cells so that the dead Cas9 could go into various places of the genome. They were going to worry about which places in the genome that dead Cas9 went after the fact. They would sequence what guide RNAs each of the cells got and figure out where dead Cas9 would have inhibited transcription. But the approach that they're taking here is a little different. First of all, they're not using dead Cas9. They're using a catalytically active Cas9. So this Cas9 will go and cut DNA where the guide RNA told them, right? And secondly, instead of flooding the cells with viruses, they used an MOI of only 0.3. Have you guys heard of MOIs before, other than when we mentioned it a few weeks ago? Multiplicity of infection, how would you describe it? Yeah, but what does that 0.3 number tell you? Per cell. So per cell, there was 0.3 viruses. What does a 0.3 virus look like? How do you infect a cell with a third of a virus? Obviously, I'm half joking. What we're trying to say here is that an MOI of 0.3, what that says is for every 10 cells, you expect 3 to be infected. So on purpose, the authors are not flooding the cells with viruses. They're expecting 3 out of 10 cells, only 30% of the cells, to be infected, right? And what these viruses are infecting cells with is the guide RNA that will go partner up with the Cas9 that these cells already express, such that the Cas9 and the guide RNA duo can go and cut DNA somewhere in the genome. And the hope is always that when that Cas9 cleaves DNA, creates a double-strand break in DNA at a specific location, when the cell tries to repair the cut, it will make some mistakes and therefore introduce a mutation. Right? So that's the thinking behind this design. My question to all of you is, why use such a low MOI? In the previous paper, by design, they had flooded the cells with viruses so that by design, they knew that each cell was going to be infected by several viruses and therefore have several guide RNAs. Why do you think, in this case, they're going for a very low MOI?
You know what I mean?
Yeah, I do. And you guys are right. You guys are correct. The idea behind this low MOI is not only not all cells will be infected, but also if the chance of a cell of being infected is only 30%, that means that the chance of a cell being infected by more than one virus is kind of very low, right? It would be 0.3 times 0.3, right? So what they're doing by design is saying, okay, I want few cells to be infected, but moreover, we want cells to be infected by only one of these guide RNAs, such that we're mutating one gene at a time. Because the risk is if you allow, by design, to have more than one guide RNA go into the cells, then chances are that that Cas9 will partner with both or more of them and cut DNA at various places, and therefore introduce several mutations, and now you lost track of what may be causing what. Because now you have several mutants per cell, and in this context, that wasn't a good idea. In the CRISPR-I context, it was because they were inhibiting enhancers, so then there is always the argument that you're not really causing all that much genetic damage, and they had that thing that by sequencing thousands and thousands of cells, they could tell when a given enhancer inhibition was truly statistically associated with a change. Here, they're taking this different approach. Cool. So the next step is to differentiate these IPSs that are now occupied with Cas9 and a given guide RNA into neurons. And for that, they carry out a protocol of neural differentiation based on the conversation before. What they do is they incubate these cells with a bunch of molecules that we know can sort of guide an IPS to become a neuron, right? And in particular, excitatory neuron. And then what they did is they incubated these neurons, many of which, not all of them, but many of which were mutant for A gene, with two fluorescent molecules. One fluorescent molecule was either monomeric or fibrillar tau. That's what we want the cells to take up, because that's what these authors were interested in, how cells take up tau from the environment. The other molecule is called transferrin, also fluorescent, but it fluoresces in a different color. And they're using it here as a positive control for endocytosis. Transferrin is a molecule that is normally endocytosed by cells, and they wanted to use it as a positive control because if a cell starts failing to uptake tau, one of the concerns may be that now this cell that has a given mutation somewhere in its genome cannot uptake anything, not just tau. So by having that positive control, they can distinguish between cells that generally can uptake things but cannot uptake tau from those that cannot uptake anything because they're dead, because they're like completely insensitive to anything outside of them and so forth. Did that make sense? So this is a classic, this experimental design is a classic experiment in which you have some sort of internal control. In this case, if you're interested in the uptake of tau, what you do is you look at the uptake of something else that will tell you, yeah, things are being endocytosed by these cells, just not tau. Okay? And how do they know when cells are uptaking transferrin and tau or either or? By looking at the fluorescence that tau and transferrin carry with them. So these are fluorescently labeled tau or transferrin. Transferrin shows up in the red channel, tau is showing up in the green channel, and essentially what they can do is they look at fluorescence in both axes. So let's say that the x-axis here is for green fluorescence, the y-axis is for red fluorescence, and therefore, what they can do is they can identify different subpopulations of cells that are taking a lot of both fluorescences. Those are healthy cells that are endocytosing everything. Cells that may be picking up zero fluorescence of either kind. So those may be dead cells or cells that are completely dormant. And then cells that may be uptaking more of one or the other. We'll go through that in a bit more detail. But after the differentiation into neurons, they would expose these cells to transferrin or tau and sort for cells or look for cells that take up transferrin but fail to take up tau, because those are the cells that they're interested in. So the last bit would be, and you notice in this cartoon there is a certain subpopulation of the cells that is sort of framed, but we'll go through that in more detail in a couple of slides. But the idea there is you would separate, you would sort the cells that are failing at taking up tau, even though they can take up transferrin just fine, and then sequence the guide RNAs that they inherited. So you take all of these cells that are failing at tau uptake, and basically you break them up, and single cell by cell you ask, what guide RNA did you get? What guide RNA did you get? What guide RNA did you get? Yeah, did you get? And by doing that, you can then identify the genes that in these cells that were failing to uptake tau, the genes that had been mutated in them, simply by looking at the guide RNAs. So if in these cells that are not taking tau well, over and over and over, you see a guide RNA for gene ABC, then you know that ABC was being most likely inhibited in those cells, and ABC now becomes a candidate for being important for tau uptake. Right? If over and over you see the guide RNA for gene XYZ, now you know that gene XYZ was likely being inhibited in these cells, some of them, and it's a good candidate for being important for tau uptake. Okay? Is the approach clear? Yeah? Okay, cool. So, before they went into this whole experiment, which is obviously very time and resources consuming, they wanted to make sure of one thing, and that is that cells could take up transferrin and tau simultaneously, and that the endocytosis of one wouldn't interfere with the other. More importantly, since they were using transferrin as a built-in or internal positive control, they wanted to make sure that transferrin did not interfere with the uptake of tau. So before going into this whole CRISPR screen thing, they took cells, they incubated them with fluorescent monomeric tau or fluorescent fibrillar tau. That's what these two plots are about. One is for monomeric, one for fibrillar. And then co-incubated those cells with larger and larger and larger amounts of fluorescent transferrin. And essentially, what they did is measure the amount of fluorescence that was going into the cells. And what you see in both graphs is that there is very little, if any, difference between the curves, meaning that the cells were able to absorb or endocytose fluorescent tau with equal efficiency, no matter how much transferrin was out there too. Does that make sense? So it's just to make sure that increasing amounts of transferrin would not inhibit the uptake of tau. By looking at the graphs, do you guys have any comments about that claim that I just made? That transferrin was not interfering with the uptake of tau? So I sort of rephrased everything that you just said there by basically asking, is this significant, right? Are these differences, could they be statistically significant? And what might bring you closer to knowing if these differences that you notice by eye might be statistically significant or not? What might be missing from these graphs that you'd be like, oh, I'd like to see that, to see if these differences matter or not? What is missing from these graphs? The one thing that sometimes you guys forget to put into your graphs, and then you get some TA or a research supervisor saying, where are the regression line, but more variation, right? And what gives you in a graph a sense for how much variation there was within an experiment? The famous error bars, right? So typically, it's totally different if a graph like this looks like this or like this, right? Those two would be completely different scenarios. In the first case, with the red error bars, I might be like, well, transferrin doesn't inhibit the uptake of tau as long as you keep your transferrin below 90 nanomolar. Because the second that you hit 90 nanomolar, it seems like tau's uptake is affected. Okay? So they didn't show any error bars as far as we know. With the, I don't know how many times they repeated these assays, but their conclusion was it's okay to co-incubate cells with transferrin and tau because the presence of transferrin is not interfering with tau's uptake. And when it comes to the monomeric form of tau, i.e. the upper graph, sure, just by looking at the graphs, I could buy that argument. Yeah? Okay. So with that, they went for the experiment. They incubated their iPSs that have Cas9 already with a library of lentiviruses containing different guide RNAs, hoping that not all cells would be infected, and the few cells that are infected by only one of these viruses, meaning they would cause one mutation in some of the cells, and then they would probe these cells or query them for their ability to uptake tau by co-incubating them with red transferrin and green tau. And then they would ask the question by fluorescence activated cell sorting, how many of these cells are taking up both, how many of them are taking up neither, and how many are taking up either of them, right? So let's say this is on purpose a blank facts plot. What you see here on the on top and on the side are the distribution of either green or red fluorescence that the researchers could see when they run the cells through the facts. So let's talk for a second about this upper graph. The upper graph refers to tau. And the way to read this distribution is that some cells expressed little, some cells had a lot. And the same applies here on this side for transferring. Some cells had very little, they're here. Some cells had a lot. Right? So that typically splits these facts profiles into generally 4 quadrants, right? So the we draw a line here and a line here. This defines, generally speaking, these 4 quadrants. So the bottom left, what does that represent? Cells that, yes. Little, right? So low. And when I say low like this, you think I'm being cute, but no, that's LO and HI is how this software for facts analysis would normally represent the different populations. Low. What about the upper right quadrant?
High.
And high, there you go. And then we would have, and I'm gonna color code it here, so we would have high for, low for tau and high and the other way around. So low and high. Right? So my question to you is, where would you look for cells that take up transferrin just fine but not tau? High, low, this guy here? The other one?
I would say the same.
You were going to say the same thing? Yeah? Yeah. You would look for cells that fall in this quadrant, that are taking up a lot of transferrin, so their endocytosis seems to be fine, but they're not taking up much tau, because that's what you're interested in. The mutation in what genes made these cells to not take up tau so well. Okay? And I answered the question myself. So what genes will these cells, the ones falling in this quadrant, help identify? So let me rephrase the question this way. So each of those cells that fall in this quadrant may have mutations in a given set of genes, right? You mutated genes randomly in cells, and now you're asking the cells, where do you fall in these quadrants? And those here would have certain mutations. So those genes that were mutated in those cells, how would you define those genes? How would you call them? What would you call those genes? Genes that are important for uptaking tau, sufficient to take up tau, important for endocytosis, important for life and viability. Like, what would you call those genes? Housekeeping genes. So the genes that we're identifying this subset would be good housekeeping genes? Is that what you're saying? Okay. Agreements, disagreements, does everybody agree? Everybody's very quiet today. I see some nodding, right? Okay, so there's still housekeeping genes. Okay, so let's turn that argument around, right? Let's say that the genes that were mutated in those cells were housekeeping genes, okay? But we mutated them. So now in the cells where housekeeping genes were mutated, will they pick up tau? No, and that's how you were reasoning before. If these are important housekeeping genes, when we mutate them, they shouldn't pick up tau. But will they pick up transferrin? Yeah? So you kill the housekeeping gene and those cells can still pick up transferrin? I'd say those cells are pretty dead, right? If you mutate a housekeeping gene, where do you think those cells will end up? In the low, right? Because those cells now are not picking up anything. What did I call them before? Most likely dead cells. So if you targeted, if your guide RNA targeted a housekeeping gene, a vital, an essential gene, most likely those ones are going to end up here, housekeeping genes, right? What about the upper right quadrant, the high fluorescence? What genes do you think you targeted there? Sort of the mirror image of housekeeping genes that, yeah. Something that is not needed, That's, never mind the introns because, we don't know where these guide RNAs were going. But clearly something, a mutation that didn't do anything. And so therefore, these cells are most likely uptaking everything because they're healthy. They can do endocytosis of both kinds, right? So these are, we would normally call those, in the context of endocytosis, we would call those inert genes. Genes that don't do anything. Now I repeat my question. What would you call those genes whose mutation made the cells end up in that upper left quadrant? Tau uptake. Yeah. Those are the tau uptake pathway genes. We don't know what they are yet, but those are related to tau uptake. Why? When we took them out, the cells could take up transferrin, but they couldn't take tau anymore as efficiently, at least. So they're alive, but for some reason they're not taking up tau. So those genes, I would call, it's a bit ironic because we're saying those cells are not taking up tau efficiently, yet we're calling those genes the tau uptake genes, and we're saying the opposite. But remember, those are the genes that were mutated and made the cells end up there. Right? So hence the counterintuition there. So those cells that we call the tau uptake pathway genes, when mutated, made the cells be much worse at uptaking tau. And we know they're not dead because they're taking up transferrin. And we know that they're failing at taking up tau because they're in that upper left quadrant where the green fluorescence is a lot less than the red fluorescence. Yeah? Cool. So that's exactly what they did. Here's the profile of cells. So some cells, you don't get a very homogeneous distribution in all four quadrants. Most of the cells fall in some sort of diagonal. Is there a question there? Ooh. Can we save that question for 2 slides from now? You'll notice that most of the cells, so these are like, those topographical maps. these curved lines is almost like looking at mountains from a top, right? So these circles here indicate that there was a peak here, and this circle here means that there was another peak here that you can imagine is coming out of the screen, right? And all that says is there were a lot of cells that were taking up both. And there were some, quite a few cells that were taking up neither, right? And then the rest of the cells, most of them fall in some sort of diagonal, meaning the more transferrin they take, the more tau they take. And the less transferrin they take, the less tau they take. That's what would make them fall in a diagonal. But they concentrated on these cells here that were significantly off that diagonal, because those are the cells that are noticeably taking a lot less tau than they take transferrin. In a way, Gabi, back to your question, I have a question here. Where would you look for cells that take up better than transferrin? You asked it yourself a minute ago. So where would you look for those? The bottom right. So by drawing a symmetrical thing, something like that, right? You're looking for cells that are off that diagonal, taking up green fluorescence better than red fluorescence, right? And they didn't do this, but if they had sorted these cells out and looked at the genes that were mutated in them, what kind of genes do you think they would have found? So we agreed that these would probably be the, I'm using Josh's words here, tau uptake pathway genes. What would those be? Okay. So Gabi here is presenting an alternative interpretation, and it's not so much that these cells were taking up tau better than transferrin. What they were doing is they were worse at taking up transferrin. So in a way, here may lie some of the transferrin uptake pathway genes. I totally agree. But what else? Tau uptake inhibitory pathway, right? So one could make the argument that if you mutated any tau uptake inhibitor genes, those cells would end up here, taking up tau better than they do take up transferrin. Right? So from a tau perspective, we can talk about transferrin some other day, but from a tau perspective, this upper left quadrant allows you to identify tau uptake inducing genes. When you mutate them, the cells are worse, are taking up tau, and the bottom right quadrant allows you to identify tau uptake inhibitory genes, because when you mutate them, now the cells are better at it. Why they didn't do this escapes my imagination. I don't know why they wouldn't have done this, because sorting these cells and sorting these cells is just equally as easy, and then the sequencing afterwards is the same. So I'm not sure why they didn't do it. Maybe we can ask, and if we have time, I have some story to tell you if we have time. All right, so they sorted those cells, they sequenced the guide RNAs that those cells contained, knowing that guide RNAs that those cells contained would probably point them to genes that were important for tau uptake, the tau uptake pathway genes, right? And they identified, remember that they did this with two different forms of tau. Sometimes they were exposing the cells to monomeric tau, sometimes they were exposing the cells to fibrillar tau, the tangles, the NFTs. So when they exposed the cells to monomeric tau, they identify these genes here. And what these plots show is when you look at all of the genes that they had guide RNAs for, some of them showed up over and over and over among the cells that were worse at picking up tau. And because you're doing something many times, and then you see a given outcome many times as well, you can run some statistics, and these genes that are color-coded purple here are the ones that passed some sort of significant test, meaning that those genes were overrepresented in the population of cells that were bad at taking up monomeric tau. And by contrast, these were genes that were overrepresented in the cells that were bad at taking up fibrillar tau. Okay? So the one thing that the authors immediately bring our attention to in the paper is that the two top hits for monomeric tau uptake are LRRK2, that's a kinase that is involved in endocytosis. So probably not a huge surprise that a kinase involved in endocytosis might be a hit. And then LRP1. Now, in the introduction, even though I failed to write it down, in the introduction I mentioned that they say that LRP1 had been found to be important for the uptake of monomeric tau. So why would they be so excited that they found something that had already been found? If you were doing these experiments and your second hit is something that had already been published, would you be happy or sad? You're not the first communicating it, you're the second to communicate that. Okay, anyone think differently? Why would you be happy? So the fact that your second top hit had already been published is validating about your approach, right? True, you'd be the second in communicating LRP1 as being important for the uptake of tau. What was this paper about? About finding new genes, right? Look at how many more you found. And basically what the authors are saying is we found, I think it's 214. Among them was LRP1, so we think our approach worked. So it's sort of validating. Let's imagine the complete opposite scenario. They do all of this, and then LRP1 is down here. Then I would stress tremendously, because it means, oh my God, my screen did not detect genes that we know are important for the uptake of monomeric tau. But no, quite the opposite. They totally hit LRP1, so they're happy. Cool, and we're not going to go through those details yet, but just from looking at some of the labels in the monomeric tau uptake pathway and the NFT uptake pathway, you can see that they're not the same. And that's one of the main messages of this paper, that these things are taking up a completely different pathway. So they had to mutate different genes for the cells to become worse at taking up either monomeric or oligomeric or fibrillar tau. So the top hit was this LRRK2 gene that was unknown to be important for uptake of monomeric tau. So as usual, whenever people run these genome-wide screens with tens of thousands of hits and candidates and whatnot, a natural next step is to confirm those findings separately. So what they did is they took cells, that's shown down here, but they took cells that are heterozygote for an LRRK2 mutation, so they have only one copy of the gene, or no copy of the gene, so they're a homozygote mutant for the gene. And then conversely, they had cells that express a variant of LRRK2 that is constitutively active, meaning this enzyme is constantly phosphorylating its targets, right? So two approaches for inhibiting LRRK2 or LRRK2, and one approach to activate it. And what they saw in the y-axis, what they're plotting is the fold change in the proportion of tau high neurons, meaning in my cultures, right, I have tau high neurons that they can take up tau just fine. Right? Tau high means a lot of fluorescence, of green fluorescence in them. What happens when I mutate one copy of the LRRK gene or two copies of it, or when I activate LRRK2, right? And what you can see is that in the controls, obviously, there is no change whatsoever, but when you inhibit LRRK2 function by taking out one copy or both, there is a significant decrease. Well, so for the heterozygotes, it was not significant, but you can see a trend. And then for the homozygote mutant, there was clearly a significant drop in the fold change in the proportion of tau high neurons. And then when they activated LRRK2 was the opposite. So now there was a significant increase in the number of cells that were taking up this fluorescence. And in both cases, I keep mentioning this fold increase. What that refers to is the fluorescence that was present in these cells before and after adding the fluorescent tau to the culture media, right? So in control cells, you see an increase that you set at 0 because that's your baseline. The mutant cells are taking up less tau, and the LRRK2 hyperactive variant is inducing more uptake. With fibrillar tau, though, the story is a little different. Neither mutation, the heterozygote or the homozygote mutation, caused any significant change. I suppose we could argue all day that there may be a trend there, but the statistical analysis said that it was not significant. But when they overexpress LRRK2, they did notice a significant increase in the uptake of the NFTs, of the fibrillar form. So LRRK2 seems to be important for bringing in monomeric tau, but it doesn't seem to matter for bringing in the fibrillar form. Okay, and that's probably why LRRK2, that's hard to say many times in a row, is not here. Right? It never showed up among the mutants that would impair the ability of cells to take up fibrillar tau, only the monomeric. Cool. Then they went for the second hit. This is, in a way, reaffirming previous observations, but they wanted to explore this a little further. So they went for the second hit for monomeric tau, that is LRP1. And what they did is, here they took sort of the mirror image approach. They basically said, okay, what is the fold change in the percent of cells that cannot take up tau or take up tau less efficiently over the ones that can? So how many more of these cells that suck at taking up tau can I see when I inhibit LRP1? Right? So that's the approach they're taking. And what they see is a significant increase in the proportion of cells that cannot take up tau. Not anything that they hadn't seen before. They're seeing it through a separate mechanism. They're taking cells, purposely inhibiting only LRP1, no more lentiviral library or anything like that. They're just introducing a guide RNA for LRP1 only in these cells. So separately, they can see that when they inhibit LRP1, there is a significant increase in the number of cells that cannot take up tau. This here is the negative control. They inhibited the expression of GFP, right? Do neurons normally express GFP? Do neurons express GFP? Yeah. GFP is something foreign that comes from jellyfish, right? So our genomes don't encode GFP. So it doesn't matter if you, quote unquote, mutate the GFP gene. We don't have a GFP gene, right? So that's a negative control, just to make sure that subjecting the cells through the process of CRISPR mutation is not affecting their ability to take up tau. So independent confirmation of what they had seen with the screen. And they went a step further and they said, okay, let's not mutate the LRP gene, but let's instead inhibit it. So they took two approaches. One was flooding the cells with a protein. They call it RAP chaperone. From what I could gather, This protein has the ability to recruit and sort of bind and hug, for lack of a better term, a bunch of membrane receptors. So if you flood cells with that, what you're doing is you're keeping receptors away from what they would normally do. And for the RAP chaperone, LRP1 is one of those receptors that it kind of binds to and hugs. So putting a lot of RAP chaperone in cells is keeping LRP1 away from its regular jobs. And same thing, they overexpress LRP1 domain 4. It's one of the domains in LRP1, the one that binds to extracellular ligands. So you can think of this as adding a lot of soluble LRP1, if you will. And what that would do is bind to ligands on the outside of cells, but keeping them away from cells, because soluble LRP1 with its ligand would probably float away as opposed to be internalized. Right? So 2 complementary approaches to basically saying, I'm not going to mutate the gene, I will simply inhibit its function. And in both cases, they saw a significant drop in the amount of monomeric tau that they could see inside of cells. So here the x-axis is tau positive objects inside of cells, and they see that the number of objects that are that have tau in them inside of cells drops significantly. Not so much when they look at fibrillar tau. For fibrillar tau, inhibiting the function of LRP1 made no difference. There was a reason why I was showing that really weird graph that was super wide with only one data point. I was masking all the other ones. So what they did is they actually confirmed several of the hits that they found in their screens, again, independently by inhibiting them separately from everything else. And pretty much in all cases, they saw various degrees, sure, but in all cases, they saw a significant increase in the number of cells that didn't take up tau so well. So the tau uptake suckers. All right? What this does is, a classical case of opening the field to a whole bunch of new research lines, right? Because now each of these could become someone's project to see how these genes contribute to uptake in tau, whether or not they can contribute to progression of AD and so forth, right? No? Okay, so last but not least, they cannot, so they had identified 200 plus here, 200 plus there. I suppose you could, if you have the time and resources, confirm each of them individually, right? But that's a lot of work and a lot of time. In the meantime, what they did is they took a bioinformatics approach, and they basically said, well, the top hits, the ones that we labeled here in these graphs, don't seem to be the same. But what if we look at all 200 plus and all 200 plus from each group? Is there any overlap, right, among the genes that we identified as being important for either taking up monomeric tau or fibrillar tau? And that's what's shown here in this Venn diagram. So the ones on the left are the ones that are important for monomeric tau uptake. Ones on the right, fibrillar tau uptake. Like I was saying before, it's 214 for monomeric, and 228 for fibrillar. Only 11 of them overlap between the two sets. And those are the 11 listed here. Only those 11 genes showed up in both sets. Okay? Is 11 a lot of genes or not? What do you guys think? No? Would that finding be, do you think that overlap is significant? You have 218 on one side or 214 on one side, 228 on the other, only 11 overlap. Is that overlap significant? Would you say, yeah, they do share a lot of genes? No? Yes? Based on what? So 0.5, so you took that route. You said, well, 200, 10 of 200, it's about 0.5%. That's a decent percentage, right? Interesting approach. Okay, you asked, like, no, that's not significant. Based on what? To the overall, right? What do they think? Was that overlap significant or not? Very significant. Very significant. So how come? Honestly, I don't know if I would call something that is 0.5% very significant, right? Like, if you're playing the lottery, 0.5%, I'm in. I'll buy a ticket, right? But if you know, for most other things, 0.5% doesn't seem like much.
This is a lot compared to the...
Oh, okay. What we're saying is that overlap, and that ties up with what I wanted to explain next. That overlap is not by chance. That overlap is not random. That these 11 genes that overlap between the two sets, it's not by chance that we see that overlap. That's what this very significant p-value says. Right? Is it many of them? No, certainly not. Right? But what the statistical test is doing is saying these 11 that you see share between the two is not by chance that they're sharing them. And where does that come from? It comes from the fact of saying, okay, given all of the genes in the human genome, what are the chances that if I pick up 11 genes from this set of 214, those are going to be also in this set of human genes? And it comes back in a way to what Jacob was saying before. These two sets are, relative to the entire human genome, pretty small. Right? How many genes in the human genome? 20K, that's right. So that's our most updated estimate is about 20K. So the idea here, and the authors conducted something called a hypergeometric test here. And the idea here is more or less as follows. If I have a sack with 20K marbles and I take 200 out of them and I look at them, then I put them back. And then I go back to the sack, shake it, and then I take out 228 marbles from that sack with 20,000 of them. What are the chances that I'm going to pick up 11 that I had already picked up in the previous, from the previous sack, right? That's what the hypergeometric test is doing. What are the chances that two sets out of a total of 20,000 would share 11? Right? So there's some math behind that, like we're not going to go there, but intuitively, what that says is the chance of these two allegedly very small sets of genes from the human genome, 200 out of 20,000 is 1 in 100. So the fact that you're sharing, and it's not 0.5%, now that on a second look, it's 5%. So the chance that you're going to get 500 or 5% overlap out of two sets that are 0.1% are pretty low, right? So that cannot be by chance. That has to be significant. And obviously, from this, the authors say these genes are not there by chance. They must matter, and they are shared by the two pathways that uptake monomeric tau or oligomeric tau.